Welcome everybody to today's lecture. So I'm the host for today. So IAS has been organizing these Nobel Prize popular science talks for a couple of years now to promote science to the general public. And so this year we're continuing that tradition, and today you're at the 2017 Chemistry Nobel Prize lecture. So just in case if you're in the wrong room, you have a chance to get out right now. And as Prudence already mentioned, today's talk will be broadcast to a local high school as well. So that would be DGS, which is a Diocesan Girls School. So do we have the video link to the girls ready? So we just wanted to see if we can say hi to them before we begin.
Hello, good afternoon, guys. In UST, we are student and teacher from DGS, and we would like to say hello to everyone in UST. Hello.
Hi. So that's Dr. Simon Orr, and he has about 118 students over there from biology and chemistry. And so after the talk, they will also be able to ask a couple of questions about the talk. All right, so I'm going to briefly introduce the talk before we actually have the talk. So today, I think everybody is in for a real treat because we're talking about cryo-EM. And this is an interesting Nobel Prize because the saying goes, this is a technology that was made by physicists, but it was a chemistry prize awarded for something to help out biologists discover something in biology. So very interesting. So today, we actually will be hearing from two biologists who use the technology extensively. One of our speakers is Professor Zhai, and she is in the Division of Life Science here at HKUST. She was born and raised in Hong Kong, but got her degrees from multiple very distinguished universities, including University of California at San Francisco for her master's, PhD from MIT, and postdoc at Stanford University. She was also professor at Cornell University for a number of years. And then the second speaker is Yuanliang Zhai, who is currently a research assistant professor working with Professor Zhai. And so they both have used the cryo-EM technology in their research, and they've published very interesting articles using that technology. So without further ado, I will ask Professor Zhai to start our talk. Please welcome her.
Thank you, Angela. Today, my colleague Yuanliang Zhai and I are very happy to share with you the excitement in the structural biology community for the award of the Chemistry Nobel Prize to the three inventors of cryo-electron microscopy. They are Jacques Dubochet, Jakob Frank, and Richard Henderson. We, like many others, are the direct beneficiaries of their inventions, allowing us to look at very large, gigantic macromolecules, biomolecules, at the resolution of near atomic or atomic resolution. Now, cryo-electron microscopy has been around for decades, but only very recently, there is a breakthrough that allows us to see these structures at the atomic resolution. And because of this breakthrough, we dubbed this breakthrough the resolution revolution, and it becomes very obvious that the impact of cryo-EM to life scientists is going to be immense. For this reason, the Nobel Committee decided that this year they're going to award the Nobel Prize to these three scientists. Okay, why do biologists care so much about structure? Structure informs function. If we can see what we work with, the molecules or the complex molecules, molecular complexes that we work with, half the battle is won for figuring out its structure. Since 1962, 36 laureates have been awarded the Nobel Prize for the development of imaging technologies for or the structural determination of biomolecules. The most notable ones in the latter category include the determination of the structure of the DNA duplex by Watson and Crick, and then the RNA polymerase, the RNA synthesizing machinery, and the ribosome, the protein synthesizing machinery. So just to give you some idea of the kind of tools that we have for determining structures at atomic resolution, I will give you a little bit of background of the limitations and advantages of each of these approaches. First, X-ray crystallography. For X-ray crystallography, the limitation is that you must have very homogeneously pure sample, such that the molecules that you're looking at can pack into tight arrangements, arrays. In other words, they can crystallize. Furthermore, these structures are static because they have to be arranged in these periodic regular arrays. And they tend to be non-physiological environments. In fact, sometimes people make mutations in order for their molecules to form crystals. The second one is nuclear magnetic resonance, NMR. For this technology, we usually use it's appropriate for very small molecules, small proteins, or protein domains, but you can look at it in solution. But the advantage of this, of NMR, is that you can look at proteins in motion, so that you can look at protein dynamics. And finally, cryogenic electron microscopy. For this approach, the beauty is that you can look at macromolecular complexes that can be as large as you like. There's almost no limitation in size. There are fewer restrictions on sample quality and quantity, and you can also look at these proteins in motion. And Dr. Zhai will talk about all these advantages and what they can do. Okay, just a few words about the range of resolution and what one can see. For optical microscopy, which has a range of 200 nanometer, one can look at cellular structures. For electron tomography, which has a range of 10 to 5 nanometer resolution, we can look at organelles. In the past, single particle cryo-electron microscopy, we can use it to look at molecular architecture because it has a resolution of 2 to 0.5 nanometers. And for crystallography, obviously it has the best resolution, 0.2 nanometers, and that allows us to look at the atomic structure of molecules. But advances have been made for all of these technologies, and I'm going to focus today on the advances that was made to single particle cryo-EM. And this only happened about 4 years ago, as I mentioned. And because of a few improvements in the use of direct detector and motion correction, one can reach to 0.3 nanometers or 3 angstroms or below. So the question is, why are biologists so excited about the resolution revolution in cryo-EM if they can see atomic structure using X-ray crystallography? So I'm going to give you some analogies so you can see how important this new technology will impact us. Imagine that you're looking at a very large molecule, a macromolecular complex, like an elephant. And it has very different structures, different parts. And if you use X-ray crystallography or NMR spectroscopy, it's analogous to looking at, because you're looking at very small parts of a large complex, is analogous to blind men looking at different parts of the elephant by feeling it. And just because you can only look at the local structure, it's very hard to reconstruct the large complex as it is, the whole complex as it is. Now, for X-ray crystallography, which has 2 angstrom resolution, it will only work if the molecule is small. It will form crystal lattice, which means regular shape conducive to packing into periodic repeating units. For example, it may be possible to crystallize the tusk of the elephant, but not the ear, because the ear is floppy, not a structure conducive to packing into repeating units that are exactly alike. As a result, many interesting structures cannot be determined because they are too floppy. Despite of all the drawbacks that I've told you about X-ray crystallography, it had a huge impact in different disciplines of science, life science, chemistry, and physical science. As you can see from the Nobel Prizes involving X-ray crystallography and structure determination since 1962. So I'm not belittling X-ray crystallography. I'm trying to tell you how important the kind of impact that cryo-EM will have. So today we are going to, Dr. Zhai will tell you about each of the contributions of the 3 Nobel laureates and how together they make this technology possible. And then he will also tell you about the latest development that allow the breakthrough.
Thank you, Big, for your wonderful opening part. I would like to start with a few examples of the high-resolution structures visualized by cryo-EM in the recent 2 to 3 years, because a picture is better than thousands of words. The first picture I want to show you is the structure of a protein complex that governs the circadian rhythm. The circadian rhythm is actually the biological clock in our body. I think some of you may know that the discovery of the molecular mechanism of the circadian rhythm was also awarded Nobel Prize this year. The second structure is a sensor that reads pressure changes in the ear and allow us to hear. The third one is the structure of the famous Zika virus. I believe that solving of this structure will certainly help the vaccine and the drug development to prevent the infection by this virus. Okay, the last one is our favorite molecules, the structure of M727 double hexamer. The M727 is the enzyme responsible for separating the duplex DNA during DNA synthesis. This structure was solved by us in 2015. With these structures and the information derived has significantly advanced our understanding of how these biomolecules operate in the cells. If without cryo-EM, these structures cannot be obtained, and we will not be able to get into the details of these biomolecules. So cryo-EM has become more and more important for biological studies. It makes much easier for life scientists to obtain high-resolution structures. So in my part of this talk, I will try my best to guide all of you through the groundbreaking discoveries made by these 3 Nobel laureates that led to the development of cryo-electron microscopy for the high-resolution structure determination of biomolecules in solution. First, let's talk about Richard Henderson. So he is the person who proved the technology's potential in determining the atomic resolution structure. Henderson was born in 1945 in Scotland. Originally, he studied physics as an undergraduate student at Edinburgh University. But later, he switched from physics into molecular biology. And he went to MRC laboratory at Cambridge, UK, as a research student to study protein structure using X-ray crystallography. So this is the first paper he published on protein structure at Nature, and later he published many other structure papers. So clearly he did a good job during his PhD study. So after receiving his PhD degree, he went on as a postdoc to Yale University to take a very challenging project. He wanted to study the membrane proteins. The membrane proteins are very difficult to manage. So if you remove the membrane proteins from its native environment, the membrane, and these proteins tend to fall apart or aggregate. So the first membrane protein that Richard Henderson worked with was difficult to produce in adequate amounts. And the second failed to crystallize. So I have to say, this was very hard for or painful for a postdoc to start with. But Henderson never gave up. After years of disappointment, he decided to switch his approach. So he turned to the only available alternative, the electron microscope. The electron microscope works similar as the light microscope. But instead of using light, the electron microscope uses the electron beam to illuminate the samples. So the electrons going through the samples will be recorded as images. So the advantage of the electron microscope is it can provide very high resolution because the wavelength of the electron is much shorter than that of the light. So the electron microscope can make very small structures visible, even the positions of the atoms. So the resolution of the electron microscope can even reach below 1 angstrom. So in theory, this was more than enough for Henderson to obtain atomic structure for his membrane proteins. But in practice, it was almost impossible because when the EM was invented in the 1930s, scientists thought it was only suitable for studying physical materials, or you can see dead material. The reason is, so here you can see the electron microscope has a huge chamber. So inside this chamber is high vacuum. So the purpose is to prevent the collision between the electron beam and the air molecules. But the vacuum is not good for biological samples. So the water surrounding the biological sample evaporates in the high vacuum. And then the biological sample will collapse. So then the sample will become useless. So another major problem for the application of EM in biological research is the radiation damage. So the intense electron beam is necessary for obtaining high-resolution images, but that will destroy biological samples because the high-energy electron can break the chemical bond. But if you use the weakened electron beam, and then the image loses its contrast and it becomes fuzzy. So almost everything indicated Henderson would fail. So if you were him at that time, what would you do? Give up? But Henderson never gave up. He was also very lucky. His project was saved by a very special membrane protein he chose to work on. So the name of this protein is bacterial rhodopsin. So it is a purple-colored protein embedded in the membrane of an organism that can utilize the energy from the sunlight for photosynthesis. So instead of removing the proteins from its membrane, Richard and his colleague took the entire purple membrane and put it under the electron microscope. And then they covered the sample surface with a glucose solution, so that protected from drying out in the vacuum. To minimize the radiation damage, they also applied a weaker electron beam to flow through the sample. So the image contrast was obviously poor. They could not see the individual proteins. But the fact is that these membrane proteins are regularly packed and oriented in the same direction. So it's just like a 2D crystal. So they can, so all these proteins diffract electrons in the similar pattern. So based on the diffraction pattern, so they can calculate a more detailed images of this membrane protein. And they also turn the membrane samples under the EM, so they can take pictures from many different angles. So in this way, they can produce a high-resolution 3D structure of this membrane protein. And they published their 3D model of this membrane protein at Nature in 1975. So this is the part of the EM density map they calculated. And here is the 3D model they reconstructed. So this was really a breakthrough at that time because so it was the best picture ever generated by electron microscope for biological samples. The people were impressed by the resolution they achieved, 7 angstrom. But this was not enough for Henderson. He was convinced that the EM can give more. So his goal is the atomic resolution that, similar to the X-ray crystallography, can achieve. So over the years, with the technology development and advancement, Henderson gradually added more details to his models. And finally, in 1990, he obtained the atomic resolution structure at 3.5 angstrom. So this was a milestone for the development of cryo-EM, because in this study, he also applied cryo technology. But here I want to remind you that so this success was built upon the special arrangement of the membrane protein. The question was whether this method could be generalized. Would it be possible to use the electron microscope to generate high-resolution 3D images of proteins that are randomly scattered in the sample and oriented in different directions? So actually, this is exactly the contribution made by Yeung Frank. So Yeung Frank made this technology generally applicable to all kinds of the proteins. Frank was born in 1940 in Germany. He is now a professor at Columbia University. So interestingly, he is not only a scientist, he is also a writer and a photographer. So he published scientific papers, stories, poems, fictions, and also photographs. This picture is one of his works that I took from his personal website. So I guess from this picture, you may have a sense that Henderson is a person full of imagination. So I guess this probably is the reason in 1975, he published a theoretical strategy for the EM data analysis. So this is the same year when Richard Henderson published his Nature paper. So in his theory, he proposed that the information obtained from the EM 2D images could be merged to generate the high-resolution 3-dimensional structure. Frank often likened his ideas to this dancing rabbit in front of a lamp. And the 2D projection of this rabbit is captured on the screen behind. So if we allow this rabbit to dance for a long enough time, and if we can capture the 2D projection of the rabbit from all kinds of angles. So using this 2D information, we can rebuild a 3D rabbit very similar to the real rabbit. So this explains his basic ideas. To get into the details of his analysis, here he called his analysis as the single particle analysis. So here we can see that the protein particles are randomly oriented. So the electron beam shines upon these particles. So these protein particles will leave their traces at this 2D EM image. And next, the computer will discriminate these traces and the fuzzy background. So placing all the similar protein traces together in the same group. So using hundreds and thousands of the similar traces, the computer can generate an averaged high-resolution 2D images. And by doing this, the different kinds of high-resolution 2D images will be generated. And then the computer continue to calculate how these different 2D images relate to each other and generate a high-resolution structure in 3D. So it sounds like this idea is not very difficult. But it took Frank over a decade to realize this idea. So in 1975, he proposed his idea. 6 years later, he published the algorithms for the software to analyze the 2D EM images. His software's name is Spider. And another 5 years later, the softwares for the 3D image reconstruction was complete. And over the years, Frank took use of his softwares and strategies to study the structure of ribosome. And he made significant contributions to the ribosome field. In fact, in 2009, the Nobel Prize in the structure and the function of ribosome unfortunately did not include him, although some people felt he should be included. But this time, he really deserved this award because his data analysis is very fundamental to the development of cryo-EM. All the advanced softwares were built upon his strategy and the principles. So next, let's talk about Yeung Zhai. He is the person who added water to electron microscopy, making the EM to be real cryo-EM. Zhai was born in 1942 in Switzerland. In 1978, Yeung Zhai was recruited to the European Molecular Biology Laboratory in Heidelberg. So his mission was to solve another EM's basic problem, how to prevent water evaporation in the vacuum. So in 1975, Richard Henderson used a glucose solution to protect his membrane from dehydrating. But his method did not work for many proteins, many water-soluble proteins. Other researchers have tried freezing the samples because ice evaporates more slowly than water. But the problem is the ice crystals disrupted the electron beam so much that the final images became useless. And then how to solve this problem? Zhai at that time saw a solution. So his idea was to make glass from water. So what does this mean? So a glass appears to be a solid material, but is actually a fluid because it has disordered molecules. So a glass is different from a crystal. In a crystal, all the molecules are arranged in an ordered manner. So when the light goes through a crystal, so the light will diffract in a certain pattern. But the glass will not do that. So that's why everyone wears a glass, not a crystal. Otherwise, you will not be able to see clearly. So the principle is also true for the ice, for the water ice. So if the water is frozen slowly, the water molecules will arrange themselves in an ordered manner. And that will form the crystalline ice. So this will blur the view. And if the water molecules are cooled down so rapidly that there is no time for water molecules to arrange them to form crystalline ice, and they will form amorphous ice. Or you can see that you make glass from water. But actually, before Zhai, many people have tried to make amorphous ice, but they all failed. How could Zhai succeed? The secret is his tricks is very simple. Instead of using liquid nitrogen, he used liquid ethane. So although the liquid nitrogen temperature is very low, about minus 200 Celsius degree, but its heat capacity is quite low. The melting point and the boiling point of nitrogen are too close. But the liquid ethane has a high heat capacity. So it's the difference between its melting point and boiling point is about 100 degrees. So that's why it can afford to produce the amorphous ice. And Zhai published his findings in 1981. He called this process as vitrification. To vitrify water, the first step is to apply the liquid samples onto the EM grid, and then remove the excess sample, just leaving a very thin layer of the sample on the EM grid. And then immediately, this EM grid is shot into the liquid ethane. So the water molecules vitrifies around the samples within the EM holes. So when the electron beam shines upon the vitrified samples, so at the vitrified water region, the electron beam will diffract evenly and produce a uniform background. So this is the device they developed it at that time for the vitrification. And they call this device as plunger for freezing. And here is today's commercial product, VitroBoard from FEI. And using their cryo technology, so Zhai and his colleagues published the first cryo-electron microscopy of the biological sample, the viruses at Nature. And from this figure, you can see that there is a sharp contrast between the virus and the background. So when this paper was published, many researchers from all over the world came to visit Zhai to learn this technique. And up to this point, the most important pieces of the cryo-EM were thus in place. And people started to combine Frank's data analysis softwares with this cryo technology. But the strange thing is the resolution remained still very poor. This situation did not change until 2013, when there was a resolution revolution taking place. So the poor resolution, at that time they called this structure as blobs, and that resolution brought this shapeless blob to atomic structure. So you may wonder, what's the problem, and what's the limit bottleneck that prevent the resolution improvement? So let's go back to 1990. So when Henderson published his atomic resolution structure. So in this paper, he discussed about these problems. So that is the specimen movement and the related image blurring. So because we are talking about the atomic resolution, so any tiny vibration of the protein particles will cause the imaging blurring. So this is just like when you take a picture of a person. While you are taking the picture, this person starts to shake his head. And the result is that you will lose the detailed information of the face of this person. And you can imagine how to restore the face of this person. So if you have a very fast camera, rather than taking a still picture, you can take a movie. So using the information from the movie, basically you can restore the face of this person. But you know, in the old days, the detector is not fast. And especially another problem with the old days detector is the inefficient electron detection. Previously, people used photographic film to detect electron. So the efficiency is about 1/3. The 1/3 of the incoming electrons can be detected. Because it's film, it cannot capture movie. So later, when the digital camera was invented and introduced in the cryo-EM, but so the electron detection efficiency become even worse. Less than 1/5 of the incoming electrons can be detected. So the breakthrough is the usage of the K2 direct electron detector. So more than half of the incoming electrons can be detected by this direct electron detector. So another important feature of this camera is it can operate in the super resolution. So when an electron hits the camera screen, so the electron signal will spread. And this camera can calculate the electron signal to identify the electron entry point. Okay, another feature of this camera is it can operate in a very fast mode. So it can capture 400 frames per second. So using the direct electron detector, you can capture movie. And then you can use the information from the movie to correct the motion. So here I want to introduce you a very important scientist, Yifan Chen from UCSF. Previously, he also gave a talk here. Basically, he's the first person introducing the K2 camera and the motion correction into the cryo-EM data analysis. Okay, from this figure, you can see that here is the position of the protein particle at the first frame. And here is the position of the protein particle at the last frame. So you can see the particle movement is very obviously and significant. Without motion correction, so the 2D image is blurring. But with the motion correction, you can see a lot of fine details of the 2D images. So using this corrected 2D images, it's very easy to reconstruct high-resolution 3D structure of the biomolecules. Another important person is Source Shares from MRC Laboratory, UK. So his contribution is to develop the more advanced and sophisticated software called Yuanliang. Yuanliang is very powerful for 3D classification. So for the biological samples, so even though you can get 100% pure samples, because the biological sample, the protein complex can assume different conformations. So in order to obtain high-resolution structure, you have to categorize different conformations into different groups. And then you can generate high-resolution structure. So here is an example. So initially, they use 130,000 protein particles. After multiple rounds of 3D classification, so in the end, they can get a subclass containing 5,000 protein particles for this special conformation. And it's just 3.8% of the initial protein particles. So use this set of data, they can reconstruct a very high resolution protein structure. So only combine all of these techniques together, the breakthrough took place at 2013 to push the resolution to the atomic atomic range for the regular protein complex. And the cryo-EM technology is still developing, and the resolution now has been pushed below 2 angstrom, and the protein size is becoming smaller. Now it's less than 100 kilodalton can be visualized by cryo-EM. So here is the figure to show you the fast growth of the high-resolution structures determined by cryo-EM just after 2013. In this year, there is already 255 high-resolution structures solved by cryo-EM. But when you compare this number with that solved by X-ray crystallography, so it is still a small number. Another reason is the high-end cryo-EM facility are very expensive. Its price tag it's not affordable for most of the university at Hong Kong. But we are fortunate. Some Nobel people at Hong Kong support our university with a huge donation that we will get this high-end cryo-EM facility very soon. So I'll stop here. Thank you for your attention. We are happy to take your questions.
All right, thank you, Professor Bick and Yuanliang, for a very interesting talk. So I think during the Q&A session, what we'd like to do is first invite the high school girls at DGS to ask their questions first. So if we could switch to the video. Hello. Hi, Simon, are you there?
Hello.
Hi.
Yes, we are in DGS.
Yeah, so we are ready to take some questions from the students if they're ready. Are there any questions?
Looks like they're a little bit shy there.
All right, so that's okay. Well, maybe we will then, why don't we kick off with a few questions from our audience over here, and if the students feel more comfortable later, let's go back over there. Okay. So are there any questions? Okay, in the back, one question up there.
Thank you for your talk. So my question is about the algorithm to analyze the protein 2D orientation you mentioned before. So you mentioned something about the fuzzy pair to identify the orientation of a protein. And so what I think is, do you think with current advancement in neural networking to identify orientations and things, will it be useful or advantageous to you apply this kind of like neural networking algorithm to the cryo-EM? Will that be an improvement into the machine itself?
Sorry, I don't know the algorithm you mentioned, but
it's just some general neural networking to look into like how the orientation are similar to each other. Will that be advantageous to the maybe like improvement of the cryo-EM? Yeah.
So this field is still developing. So people are trying now. More mathematicians jump in this field to develop much better algorithms. I agree that better algorithm will be developed very soon for the 2D data analysis. Yeah.
Any other questions? Yes. Oh, could you please use the microphone? Thank you.
What is the current detector used in the machine, the commercial machines?
You mean the
Yes.
The one I shoot here?
The one that costs a lot of money.
There are two brands of the direct electron detector. One is from FEI, the name is Falcon. The other one is from Gantan. The name is the K2 Summit.
Yeah, what are they?
Both of them can be used.
What kind of detector are they?
The direct electron
detector. Silicon detector or CCD detector? Or what
silicon detector. CMOS.
It's a CMOS detector.
CCD, I think.
No, CCD is...
Sorry, did that answer your question?
No.
Okay.
What is the detector used?
Yuanliang knows. What is it? Wang Ning, I mean, Wang Ning.
Not CCD, it's a CMOS. It's originally CCD.
Use a microphone.
Using all right. I just try to explain it, but since we know a little bit about this detector, previously people use phosphor and glass fiber to transmit image directly on a CCD detector. And then people think about whether we can detect electrons directly. And the only problem is that the high energy electrons destroy the pixels very quickly. Okay, and later for the new technology, I think people already use CMOS technology, the pixel functions to directly detect the electrons. But the coat a very thin layer to minimize the damage of electrons on the sensors. Basically, the sensors is old technology. It's not new. Thank you.
Okay. All right, there was a question up the front.
Oh, actually, it's a comment. Because I got to know this method about 10-some years ago, tried myself, and now Yuanliang has just showed the publication rate increasing exponentially. To me, yeah. If the Hong Kong USD doesn't get this equipment very soon, I actually suggest not to get it. It's too late. We talked about like a couple years ago. Now, like Beida, Tsinghua has 5 or 3. Beida has many. If let it go for another year, I don't think we can catch up because we want to do high impact research. So maybe for 100 people. But this, if we want to have it, we must have it as soon as possible. Otherwise, we just follow other people. We can never catch up.
Okay. Very lucky for us, we are in the process of ordering one such electron microscope, the latest. So it will happen soon, but there's a back order. So we have to wait in line. But I want to add that in order to be successful in solving structures in cryo-EM, it's not just the machine. As you say, there are universities that have 4 sets and other universities have 5 sets, but they don't necessarily publish high-impact structures or structures that are really good. The important part is sample preparation still. You have to be able to purify, especially you get larger and larger, more complex molecules. They are harder to purify, and to get large quantities is difficult. So the sample preparation is very important. Also, the grid preparation is also there are a lot of tricks and different molecules you're required to do different things. It's not a formula that one can use in terms of sample preparation. So we want to beat others. We just have to be better in sample preparation and grid preparation.
Okay.
We're not behind.
No, I don't think so.
Yeah, we're sending people to Beijing to use their facility.
Yes.
All right, so I hear that the students at DGS have some questions also, so maybe we can take a few from them. Hello? Okay, we can see you.
Professor Tai, Professor Wu, and Dr. Zhai, thank you for your inspirational speech. As we hear just now, the biomolecules are frozen when they are being studied. Is there a way to use this technology to visualize how they undergo conformational changes when they're in action? Thank you.
Okay, shall I? All right, so Dr. Zhai mentioned that when you have a sample, the molecules can be moving, and you can categorize the different conformations in the different modes of action. This is what the 2D and 3D classification is about. So if you can classify these different conformations, and in a way you're making a movies also of the different conformation at different stages, and then from those different classes, you can deduce how the molecule moves, how the complex moves. Have I answered your question?
Thank you for answering your questions.
Thank you for your question. Any others from students over there?
Maybe I would like to ask a question myself as well.
Of course.
Since maybe our students are really a little bit new to that topic. And then I am personally interested about how the technology can help us to find out interaction among different substances. Is it true that we can only work on pure substances like a single protein? Or is it possible to have more than one, so we can see how the image of these proteins when they're interacting, or when they, for example, different subunits of it, when they combine together, would there be any kind of help in solving this structure determination part? Thank you.
Thanks. Thank you.
Okay, so the whole thing is about single particle analysis. If you have in your solution too many different molecules or different, then you will end up with a lot of noise. You don't know what you're really looking at. So there is still some kind of limitation in terms of your samples still have to be quite homogeneous. But we were trying to, as I tried to answer the question before, we are trying to capture the motion of something quite homogeneously pure, but that doesn't have to be completely homogeneously pure because we are not crystallizing. We're still just counting, looking at single molecule images, single particle images. So the technology is really geared towards looking at a complex that may be undergoing different conformational changes, but not. So I think we're still far from. I think there'll be too much noise if you have too many different molecules in the same sample. You'll end up with noises.
Yeah, maybe I can add one point. If you combine A or B together, just two proteins or two complexes, that should be able to visualized by Cryo-EM. Because nowadays the softwares are very powerful to do the classification. Yeah.
Does that answer your question, Simon?
Yes, sure.
Okay.
Thank you.
Thank you. And maybe one last chance. Are there any other students over there that would like to ask a question? Okay, maybe not. All right, so if there are any other remaining questions in the room, we can take those. Yes.
Okay, thank Big and Yuanliang for a very nice talk. I learned a lot. So just a small question. I was curious about the second example you gave for the structure. So you said there's a sensor for in the inner ear. So what is the protein you're talking about?
I cannot remember the name, but that was solved by Yifan Chen's lab. Yeah, published very recently in Nature.
Okay, yeah. So it's I think it's the NOP-C. But I don't think it's responsible for hearing in the inner ear, because that's something I'm working on. If it's already solved, so I'm out of a job. Okay, so it's not exactly right. Yeah, it's some kind of sensor, but not in the.
You can purify the complex.
Maybe in the Drosophila, it's not in Drosophila. Yeah, it's not in human. Yes, you're right. Okay.
All right. Thank you. Okay. I think if there are other questions, we can take one last one. If not, if there are no more questions, then let's give a round of applause to our speakers one more time. Thank them for giving us a very interesting talk.
Thank you.
And thank you for coming. Yeah, and maybe let's say goodbye to the girls over there. Bye.
Bye.
Thanks for joining us.
Thank you for joining us.